workshop with the first speaker, Dr. Rajesh Karyakarte. And as I just mentioned that he's the best, he won the Best Teacher Award by DMER. Now you'll exactly understand why he got that Best Teacher Award. And it would be a very beautiful talk by him. He will just swim through the complete sequencing in general, and he'll cover the complete sequencing. I think whoever wants to go home can go after this talk, except if you don't want any hands-on.
Let me first invite the VIPs here. The Dean himself wants to know about genomic sequencing, so he is amongst you. Dr. Tambe. He is going to talk on molecular epidemiology, as we have discussed now, to make a policy decision with Government of India, because they have a conference of, it's a national conference of PSM, which he is going to talk on there. He is going to talk on the policy decisions which can be taken for the theme of this particular conference, that is molecular epidemiology. And I have the privilege to have the youngest of all students with me, Dr. Jagirdar. So yeah, so yes, so I thank you very much, because see, it takes a great guts and courage for the doyen of this field to commit in front of everybody that, well, he doesn't know something which the person has to be well grounded and at the top at the same time to say something like this. So thank you, sir. That is really fantastic. I will start in a very unconventional way, but the main aim would be to take you ultimately to the end on a very, what should I say, a very well smooth drive through the whole process. Now, why genome sequencing, and what is this genome sequencing? For that, the first question which should come to our mind is, what is life? So it's a philosophical question. I will come. And the slides I will show afterwards. I like to talk first. So I will first talk with you all, and then we'll see the slides. We'll have a follow through of the slides later on. So please listen to me what I'm telling you. So the question which comes to our mind, a philosophical question which comes always to our mind, is what is life? There are many people who have talked about life and all those things, but we scientists, we have very simple understanding of what life is. Life is DNA, RNA and protein. That's life. Because we are made up of proteins, and the DNA is the one which is keeping all the information, and RNA is one which is transcribing from the DNA to the protein. So this is life. This is life for the virus also. And virus is a bit different from us. So we have viruses which have a genome which is an RNA genome. We also have certain viruses where the genome is RNA, but it first forms a DNA, and then the whole thing comes again in that particular loop, that is DNA, RNA and protein. Well, we have this particular SARS-CoV-2 which is of interest for us today, and that particular virus, it is a RNA virus, a positive sense RNA virus. So it can act as the RNA, mRNA itself. So it can form its own protein straight away without depending on almost anything except its own polymerase. That's the only thing which it would need. So if this thing is very clear to us, we should go a bit further of why this sequencing should be important for us. The sequencing should be important for us because, or I would ask another question. The first question I asked was, what is life? The second question which can I ask is, what is the language of life? What is the language of this life? The language, we have, for example, we have English, we have 26 alphabets in it, and all the words, maybe those words some are very long words like actinocomitans, right? Actinobacillus, actinocomitans. It's almost 25 or 30 words, alphabets in length. Then we have I, only one alphabet. Me, 2 alphabets. But then the life, the code of life, there are only 3-letter words there. You will like 4-letter words, okay? But then you have only 3-letter words here. And these 3-letter words are called the triplet code. So we have a triplet code, which is a degenerate code as we all know it, because the whole code represents for 21 amino acids, which we can form. We can produce those amino acids. The viruses also do the same thing. They go on producing amino acids from their particular triplet code. The virus has something very special, and that special is that we have a gene. A gene is one protein. That is what we are taught. That is what a eukaryotic genome says. That one gene, one protein. But not with viruses. They can have a single gene and then they can make many proteins from it. They just change the reference frame. The reference frame is changed, you can have a different protein from it. But in any case, you are going to have a triplet code. So 3 nucleic acids are going to code for one protein. This is clear for us now. I am now moving forward. Now why is this important? It's important because if you have a point mutation, if you have a mutation, single nucleotide mutation, what can happen is, you can have a different protein being formed. Now this is very critical. This is critical in the case of SARS-CoV-2, this is critical everywhere. Now what is the criticality there? The most important thing which all microbiologists know is that antibodies recognize shapes. Antibodies don't recognize chemical formula. They recognize the shapes. And what determines the shapes? All the amino acids determine the shape. So if you change even a single amino acid, you can change an amino acid from a hydrophobic amino acid to a hydrophilic amino acid. You can change an aromatic amino acid to an aliphatic amino acid. You can change a negative polar amino acid to a positive amino acid. And what happens because of that? Just try to remember what happens if a particular charged thing is, or a hydrophilic or a hydrophobic thing is in water. What will happen? All our membranes are formed because of this particular characteristic. And that is, if something is hydrophilic, it tries to come on the outside in touch with the water. And if something is hydrophobic, it tries to go inside. Now this particular fact, try to understand it very clearly. What would happen? The thing that would happen is, if you have a protein where something is hydrophilic and is projecting out, so what is happening is, your immune system is recognizing those epitopes as immunogenic, immunodominant, and your antibody responses against those which are on the surface. So all the protein, I'm talking about humoral response. I'm talking about humoral response. The antibody response to these particular proteins would be based on what is on the surface. So if anything is inside the virus, it's not going to be that immunogenic to antibodies. So we all know that the spike protein of SARS-CoV-2 is immunogenic. Why? Because that particular protein is on the outside. It's exposed to the outside. It is the one which binds with the ACE2 receptor. And therefore, that's the most important part of that particular virus from immunological point of view, particularly humoral immunity point of view. The second concept which we have to understand very clearly is that it is the shapes which are recognized by our immune system and not the chemical formula. If you remember Anand, I shouldn't tell this to our teachers, but there it has very clearly said that the site chains, if they change they change the whole shape of the antigen, and therefore the antibodies are produced against those shapes. Now just try to understand what would happen. What would happen to a spike protein if a particular hydrophilic amino acid is changed to a hydrophobic amino acid? The moment that happens, a shape which is projecting out comes in, caves in, and the antibody which was formed against that particular domain, or that particular antigenic determinant, or that particular epitope is now useless. This is precisely what has happened with SARS-CoV-2. All the major mutants, all the major variants have produced mutations which have changed the shape of your spike protein, and because of this change, they have gained the property called immune evasion. So when we talk of all, we talk of Delta, we talk of Kappa, we talk of Omicron, we talk of BA2, we talk of BA1, we talk of the latest which we have found out, BA.2.75, all of them produce mutations which changes the amino acid, which changes the shape, and therefore changes the structure. Now here comes the importance of genome sequencing. You have to catch that particular variant early. And how are you going to catch it? You are going to catch it because if there is a mutation, there is a mutational difference in maybe 4 or 5 such isolates, we can think that something new is coming. This we identified in June itself. There's something new is coming on, and that new was BA.2.75. And today it is dominating the whole world. This was done 2 months back. So that is what gives this sequencing an advantage. We did a very fast study with that. We did a fast study of clinical study for that. I have a fantastic team which works on it. So I had 20 postgraduate students asking questions to all the PhD patients of BA.2.75. And we came up with a data which told that this particular organism is not causing deaths, not increasing the oxygen demand, nobody is going to ICUs, so need not fear that much as we had to fear Delta. So there's nothing new Delta coming up. That was the thing which we could tell based on this molecular immunology. So that is the importance of molecular immunology, where you can predict what is going to happen tomorrow in the world. I'm not talking of India. In the world, what is going to happen. And the prediction right now is we are going to have BA.2.75 all over the world, particularly BA.2.75.2 all over the world. You can read the newspapers maybe a month after and you will get the answer. So that insight we can get because of genomic sequencing. So I think I have sufficiently emphasized on you what is the importance of the genomic sequencing. One more very big thing which could tell all the whole world is that BA.2.75 is dominating BA.5. This is what we told the world. And that is a very important thing because BA.5 is killing a lot in USA and other countries. And it would be replaced by this milder variety. Maybe there would be some succor for them. That is what we are hoping. I don't know what would exactly happen tomorrow, but BA.5 is on the way out. Because of the sequencing studies we have made at BJ, we can predict this for the whole world. And I think that is something which every one of you should understand, that in your labs, in your colleges, you can do the same work for SARS-CoV-2 as well as all other bacteria and viruses, particularly tuberculosis. It's very important for tuberculosis and others. So I want to demystify the whole sequencing thing for you. That is my job. That was my idea behind this hands-on workshop which we are having today. I hope that is clear now. Now let us move to the sequencing technologies. What is a sequence? A sequence is, what should I say, a single line made up of nucleotides. Now what the bioinformaticians have done is a very easy thing. Each of them is an alphabet. So A, T, C and G are, for example, the alphabets. You write them from the first alphabet to the last alphabet, all in A, T, C, G. The whole thing is your genetic code. And you can very easily write it on a piece of paper. You can write it on a simple text file. So the whole thing which comes on that text file is called a FASTA file. I think now things should go in. Things should go in now. That is, we have something in bioinformatics which is called a FASTA file. This has to be understood. And this is a very simple thing because it is just a text file which can be opened by a notepad. I think everybody knows about all the computer programs, so I think it is clear to you now that it is a notepad file which can be opened simply, and just you will have to then compare two different sequences. And if the original sequence, for example Wuhan string, has a particular sequence and amino acids at a particular point, and then your isolate has different nucleic acid at a particular point, you say that there is mutation. Then you see for the changes in amino acids there, and then you can predict whether it has an immune escape function, if that particular amino acid is on the top, on the outside of the virus, or if it's inside the virus, may have some effect on the cellular immunity, we don't know, or pathogenicity, we don't know, or it may kill more, if people may need more oxygen. That all things can be predicted from the structure of the protein which is affected by this particular mutation. So that thing is also now very clear. But then, to make such a simple text file, you will have to use certain technologies which would somehow identify that nucleic acid and convert it into this text file. So how are we going to do that? 3 companies, some are sitting here. What can they do? They can do certain things by which we can get to know what that particular sequence is. How do we do it? We do it with the Sanger sequencing. For example, you have a fluorescent dye which is attached to all nucleotides which are of different color, and then see what is the fluorescence which comes up in each of this step when we are adding those particular nucleic acid. If suppose adenine with this fluorescence is taken up and it fluoresces that particular color, then we can say that adenine has come in that sequence. So, all these things, I think you're getting the point. So it's the fluorescence which can be seen. There's one more technology called Illumina technology, where they can just photograph the whole thing. The color can be photographed, and then they can have a file on basis of that. You have ion torrent technology, where they can just find the release of hydrogen ion each time a bond is formed between two different nucleic acids. And then you have something very special, and that is a nanopore technology, where something goes, there is a pore there, something goes through the pore, and while it is going through, you have something very simple device there. You have a membrane, an impermeable membrane between water. For example, you have a trough, for example, in which you have water, and you have separated it by the help of a membrane which is totally impervious to water. So, and you apply electric current on both sides. Now, what would happen is, because it is impervious, no electrolytes would move through, and you won't get any current at all. Now, if you make pores in it, which are called nanopores, of very small dimensions, through which a single molecule can go through. What would happen then? The moment such pores are made, electrolytes can freely move through that particular fluid. And if you apply electric current, the current would pass because of the electrolytes. So you would get a current there. So things are clear. If you have something which is impermeable, you are not going to get a current. And if something has holes inside it, so that the electrolytes can move easily and freely through it, you will be having current. Now think of a case where you have a sequence of amino acid, which we, by different procedures which were told to you on the video, we put a particular strand of DNA through that particular pores. All the four of them, the four nucleic acid, have different sizes and they have different charge. So when they're going through, they would be blocking that pore at a different extent. And the electrolytes which would pass through would be proportionally less or more, depending on its size. So there is going to be a small change in current each time a nucleic acid passes through. And that change in current, how much it has dropped, for how long it has dropped, is going to tell you about which nucleic acid has passed through. Now this, whatever is happening, is a machine reading. So what is the machine reading here? It's reading the electrical currents. And when it makes a file out of it, it's called a fast 5 file. I hope it's clear now. We had something fast A, I'm moving towards fast A now. Right? So we started with fast 5. It's an electrical current which is taken, and all the changes in electric current are noted in a particular file. It's a binary file. It just electrical current 0, 1, 0, 1, 0, 1. That is how it records it. Human beings cannot read it. So it is read only by machines. And that is what is called as the fast 5 file. So we have first thing which happens there is fast 5 files. In the case of Illumina, it's just the photographs which are there. In Illumina technology, it is photographs which are there. In ion torrent technology, it is the hydrogen bond and then again the electric currents which are produced there. Now once you have this fast 5 files, we have software which are based on artificial intelligence and machine learning, which can be trained, and they come to know that if the current is fallen for a particular time and a particular drop is there, this one is adenine, the other is cytosine, other is thymine, and so on. So when a software, Guppy for example, it is analyzing these signals and writing another file, which now is in the form of ATCG, but not a text file as such, because it is also giving quality scores to it. Quality scores means how confident that particular machine, that software is, to say that it is A, to say it is G, or say it's T or maybe C, something like that. So how confident it is done in form of scores, quality scores, which are called as spread scores. That is how it happens. So I hope you are getting the whole thing now. You have a software which is going to analyze this FAST5 files, all the electrical signals which are in form of zeros and ones, into now a nucleic acid. And while it's doing so, it is also writing a quality score for each of that nucleotide, of how confident that software is to say that it's A. Here comes the role of consensus sequencing. Now what is this consensus sequencing? What happens is, it's not just one molecule of one particular segment which goes through the pores. There are thousands of pores through which it goes in. So when it's going through, each time maybe a single particular sequence will be going through. So the same sequence maybe goes for 1,000 times. So when that does that, we say that the depth of sequencing is good. So it becomes very confident in saying that, well, this is A, because I have seen it for 1,000 times, and in 999 times it was A. Maybe for one time it wasn't. But then I am confident at this Q score that, well, this has to be A. So this is something called as consensus sequencing. But ultimately, let us try to understand it later also, because Dr. Rashmita will deal with it something, or maybe I have to do with it. But then, yeah, then what happens is, you get a new file now. See, try to understand. It was FAST5 file to begin with. This, a software like Guppy, takes up the whole thing and writes it in form of A, P, C, G, and gives a quality score for each of them. This whole thing is called as a FAST-Q file. I'm telling bioinformatics to you. The lecture which was going to be taken later on. So it's FAST-Q file. So what did we have to begin with? We had a FAST5 files. It was machine readable. It was just zeros and ones. Then we have FAST-Q files, which is done again by software, machine learning, and artificial intelligence. And it tells you that these are the bases. It tells that it is the score of the bases also. It tells you a FAST-Q file is written. This FAST-Q file is then converted by other softwares into a FAST-A file. I hope you get the point now. Ultimately, what did you want? You wanted a text file which is called as FAST-A file. So we have got it now. In the case of Illumina and other technologies, the same thing is followed. But those are image files there because they are looking for color and intensities. Here we are seeing for electrical fluctuations. Right? So technologies are different, but then the main aim of all the technologies is going to be to form FAST-A files, because they can be compared by different softwares which are available all around the world, luckily free of cost, and that can be done. So right now what we are using is 3 different softwares. Those softwares are available free on internet. One is Nextclade. This one gives you different clades. And let me tell you, I was talking about BA.2.75. This lab's sequences were utilized by Nextclade to give a new name to BA.2.75, and that is 22D. So the 22D, guys, 22D is our genesis, BJ Medical College's genesis. So the name which is given to that particular virus, the Omicron 22D by Nextclade, is based on our sequences, and the certificates which you are going to get has it as a watermark on the left side. And that is done with a purpose, right? You just have a look at it and you will read something called as BJ-COG. That BJ-COG is this fellow and this thing here. So that is how we have been instrumental, and it can be done by each one of you the moment you get it, get the technology in your hand. And important thing is you want to use it, and you want to use it for the betterment of mankind. That is the main thing, that is the jazba which you should have. Unless that, then the sequencing is very difficult otherwise. But if you have this jazba, I think sequencing is very easy for you. So well, that is one thing. The second one is a Pango website. The Pango website gives you the lineages. Like for example, BA.2.75 or BA.1.1.529. This is the Omicron variant. Or BA.1.617, that is the Delta. So these names are given by Pango. And there's a group of scientists who work on it and give different names. And the third one is Gized. Gized also gives its name, like that G variant or this goes on giving names in that particular fashion. And lastly, the last fellow which came about was the WHO. They thought that this BA thing is getting very difficult. Maybe this 22D, 21D is not beyond comprehension. And people, particularly the press, needs to know, have a name which is understandable. That's what they think, but I don't know how we can understand alpha, beta and delta. We don't use those words. E would have been better for us. But then, well, they say that it's the Greek and Latin and something like that. And they say that, well, the first one which came about, which was different, which was VOC variant of cancer, is alpha. Then they named it another one as beta. Then delta, we named it. We named it, we found it out, but they named it delta. They named it kappa. And so on. Now they have Omicron. And in the meantime, they left the Chinese thing behind because those alphabets are not taken. Not taken, right? So the politics also there. So yeah, so those things also happen, and we ultimately have those names to us, names with us. So I think I have told you the entire flow of the sequencing. We have understood it now. We have understood it now, right? Sequencing is for knowing the mutational changes. The mutational changes can be easily found out because ultimately what we get is a FASTA file. FASTA file is just one particular sentence which is telling us from A to Z. How many words would be there for this SARS-CoV-2? How many words? How many words? How many words? We have to know that. What is it? How big is its genome? How many bases there? It's 30K, 30,000. So the sentence which is written by SARS-CoV-2 is 30,000 alphabet long. 30,000 alphabet long. So that is how the whole thing is. And we just have to compare it with the other things. And that's the thing which you get. And then I told you how to reach this FASTA file. I told you that we have to go through the FAST5 file first, where it's just a machine readable file, and then we need a software which is based on artificial intelligence, machine learning, which is going to tell you that this particular 0101 is meaning A, B, not B, sorry, A, T, C, G. That is how it is naming, and then ultimately you get the FAST5 file, which can be compared with the original Wuhan strain virus, and then you can identify that particular variant. With this talk, now let's see fast of what I'm trying to show you, and we'll finish the whole thing off. Yeah, so my basic question. Hey, but I like to ask this question because I am prevented from going to Himalayas. What is life? So, what is life? Life is simple: DNA, RNA, protein. Sometimes RNA codes for DNA. All our microbiologists here. So, for the benefit of non-microbiologists sitting here, can you please tell me what is that particular virus called, where it's an RNA virus and then makes a DNA out of it? Anybody? You have to be with me, yeah? What is it called? What is that particular family of viruses called? Retroviruses. Yeah, sir त्याला retrovirus म्हणतात. आणि HIV त्याचा एक भाग आहे. HIV व्हायरस हा retrovirus आहे, जो RNA व्हायरस आहे, पण तो आधी DNA करतो आणि मग जे आहे तो हे. आणि retrovirusचं महत्त्व असं आहे की retroviruses are present can cause cancer. It's only RNA virus which can cause cancer because it can integrate in your DNA. So this is life. Sometimes it can start from RNA itself. And that is all RNA viruses start with RNA. They don't have a DNA there. So yeah, this is what is there. Okay, this is the building ATCG, and in RNA, a U comes in place of AT. Then these are the base pairing occurs. It's very easy to remember: CGAT, at center of gravity. This is a strand. This is a sequence. This is a sequence. The triplet code. Now this is a very silly thing here. I would want you to see. Artificial intelligence, what can artificial intelligence do? This is a suggestion which is given by PowerPoint presentation. The moment I wrote code, it gave a computer code in the background. So, machine learning can go wrong. That is what I want to tell you. Yeah, this is the triplet code. You start from 5' end to the 3'. So, you can see that there is a start codon. Start codon is a constant codon, the met one. And there are 3 stop codons. So, the punctuation is very simple in the language of life. You have no commas there. You don't have any semicolons there. You start from something and you stop it. That's all. So when you stop, you form a protein. That is the end of the protein. So this is the triplet code. Do amino acids love or hate water? They certainly do. Because you have hydrophilic amino acids and hydrophobic amino acids. This is how it is. This is the entire list. The polar, non-polar, positive charge and negative charge. Please have a good look at it because something very remarkable is going to come on the next slides. So this is very important to understand. That amino acids, all amino acids are not same. They are different. Some have a polar group, some don't have. Some are acidic, some are basic. And also something more. Something is small, something is tiny. Right? So this is the amount of variation these amino acids have. So obviously if the amino acid is a tiny one, the shape of that particular antigen, that epitope would change. So I want you to understand this thing that the entire plane is of the amino acids which make up the protein. And that is by the genetic code. So single alphabet change can lead to some other amino acid and then a different shape and therefore immune escape. This has to be understood very clearly and that's why we need sequencing. What happens when how do proteins fold? And when they fold, what happens is, all the hydrophobic groups go to the inside because they hate water. They don't want to be in contact with water. And all hydrophilic things come on the outside. And they are the ones which are exposed to the exterior in aqueous environment. As you know, we are almost 90% water. We can have, we can talk to each other. We can have a question and answer session unless it comes in. Oh. Yeah, right now we have a recombinants of BA.1, BA.2. We have a combinations of AY series. Now what is the AY series? They were the further lineages of Delta, so AY. Right now we have the BA series of Omicron. So they can combine and they can form a new virus. Luckily up till now what has happened is that all those viruses have not been more pathogenic than the original or more than the Delta. And that is the safety concern.
So variants may not survive also in nature.
Yeah, they don't survive. The survival of the virus is very easy and to understand. I'm just digressing a bit because things were coming up. One thing which we have to understand very clearly is a simple epidemiological fact. And that simple epidemiological fact is a person who is seriously ill stays on the bed and has little chance to infect others. On the other hand, a very mild virus, the person himself doesn't know that he is infected. He goes on sneezing and talking loudly everywhere and goes on infecting everybody. So obviously, a pathogen which is milder has more chance to spread than a pathogen which is serious. Serious pathogen would bedridden a person. So he doesn't have a chance to spread the disease. So that is how even when many people are saying that it's not true, we have seen it in the corona that it is true, and that is the virus has become slowly and steadily milder and milder and milder. Two things there, the virus may or may not have become milder, but we have an immune wall of our own now due to vaccination as well as repeated infections which we have. Okay, now it's back now. So yeah, this folding is very important. It's important here now. These are the mutations of all known variants. So variants show mutations. They show mutations particularly in the receptor binding proteins and they also show mutation on the exposed S protein which we have, and therefore they gain an immune escape mechanism, and more importantly, they can also gain a mechanism by which they can infect the cells commonly. They can infect the cells more efficiently. They can spread from a cell to cell more efficiently. Tropism is also determined by these mutations in the sense that you can have, for example, Omicron causing infection in upper respiratory tract rather than lower respiratory tract. So all these things are by these particular mutations. Now I'd like to show something very remarkable as we move forward. This is the alpha and the Omicron which had the deletion of two different amino acids at the position 69 and 70. Now what has happened there is that the S1, S2 lineage and spike incorporation and the infectivity and the fusionity changed because of this deletion of two different, with six nucleic acids were just deleted out. So two amino acids just didn't happen there. Two amino acids not there at all in that particular protein. And then what happens? You get these increased properties of that particular variant. Now let us try to see that data. I have purposefully used the colors. I purposefully used the colors somewhere here. I will show you. I have used these colors. I have used these colors there to denote that the yellow thing is non-polar and hydrophobic. The blue one is polar and hydrophilic, loving water. Then you have a positive charged one, which is hydrophilic again, and a negative charge. So these colors, just now have a look at these colors and try to recollect what I am showing you now. I am showing you something here. And if you see carefully, the colors are not same at all after mutation. These are very significant mutations of delta. Particularly, you have L452R, which is the most important mutation of delta, where we have a fear that this one is going to cause problems with BA.5 and BA.4, which has this particular mutation. So this is that important mutation. If you see it carefully, a hydrophobic thing has become a hydrophilic thing. Now what has happened is that that particular place, which is inside a structure, has now come on the outside. So you would think that something, if it had come outside, projecting out, maybe it's dangerous. No, it's not dangerous in the sense that the antibody which was there covering that particular region was looking for a khanda there. It didn't have it. He had a dongar there. So how can it bind? It just cannot bind. Because you know, antigen antibody reactions are best when there is a close fit. So if there is no close fit, the antibodies are not going to bind. You are going to get an immune escape. So I think you understand it very clearly now. So if you see carefully, none of the colors are same. So a hydrophobic thing is changed into an hydrophilic thing and vice versa. Right? With Omicron, something different has happened. It has kept this trend, but something different has also happened here. So if you see very carefully, N501Y, and this is a defining mutation for Omicron, N501Y. Colors are same, but if you go to the depths of it, the sizes are different, and the side chains are also different. So what is happening is, again you are getting an immune escape problem. So the virus has found out the simplest of way to evade our antibodies. Maybe it has to come to know that these fellows can identify me by shapes only. And the only thing I have to do is to change my shape. So I think that is the thing which, that is the message which we get of why this virus is not under control still. Yeah. These are the steps in sequencing which would be covered. So I'm just going through it very fast. And I think this was told in that video also. Sorry. Just to recapitulate, or just to tell you, if you want to find a sequence of a virus, you will need the virus in the specimen. This has to be understood very clearly. So unless you get a positive sample, you are not going to sequence anything. Further, the amount of the virus in that particular specimen is going to be determined by the CT value of that particular sample. If you have a very small amount of sample, it would come positive in maybe 30, 32, 33, but that doesn't help you. You need a positive sample less than 25. So unless you get that, you don't have enough RNA in it to do sequencing. Sequencing is very costly. According to Instacog, one sequencing takes 5,000 rupees. So, and this PCR takes maybe 20 rupees right now. So PCR is better. Do PCR and see what is happening. So that is how you get the things done. I hope you understand. So why the sample is important? Why the sample has to be transported with care? Because RNA is very labile. If it was so strong, we wouldn't have DNA. Our genome would have been RNA. But it's not our RNA. Why is it not an RNA? Because RNA is weaker than DNA. So if you are going to transport DNA in a temperature which is 25 degrees centigrade, you are going to lose most of the RNA. You are going to lose it. Then, even if there are traces of RNases in that particular sample, you are not going to do anything. For example, you use that antigen test where you destroy the whole virus, and then you don't have any RNases there. That sample would be broken down, and if you use that particular sample, maybe you won't get the run, and therefore what you need is you need a RT-PCR sample, that is sample in medium. It's something which I have already touched. You see that particular thing which is very black there, that is the nanopore, and the orange thing there is the motor protein. So obviously the motor protein is the one which is pushing that particular DNA through. So you should understand it very clearly that if you have to do that, you will have to have a process, a step where you are going to attach this motor protein at end of that particular sequence. So you are going to have one step where this particular thing of binding is going to take place. First thing is you are going to get your RNA, which you are going to convert into cDNA, and then make small pieces of that cDNA of specific sizes, 1,200 base pair, and to them you are going to attach all these things, and you are going to attach some other things to the surface of that particular membrane so that it tethers it, so that it catches it up. The motor protein is brought to that particular place where you have the hole there, and the motor protein uses ATP, glucose, to run. So your reagents need to have glucose, need to have ATP, need to have other things which are needed. So that would be the exact process which is told by Dr. Shweta, how it happens. What do you have to do to see that this thing happens actually? And then as it is moving through that particular pore, you are going to get blockages there. So the electrolytes are going to be less going through, and therefore the current is going to change. And current is going to change for some time and some depth. From that we are going to identify the. So this is how it goes through. It's called translocation. These are the machines. These, the MinION and GridION is available at our institution. This is in short those steps which I told you. We're going to be dealt with in details later on. You can just have a look at it. I've added all the steps. Step 1, reverse transcription. So you get cDNA. Then we add rapid bar. This point needs to be emphasized. This is a very costly procedure. So it's better that you do it in one go for many samples. So if you want to do 96 samples at a time, then you need to identify each DNA as belonging to a particular virus. So what we will have to do is we will have to barcode it. The barcoding, to understand it, that's very simple. That is, there are stretches of some 20 nucleotides which go and bind to either end. If a machine reads that this is the nucleotide which was stuck there, we call it barcode 1. Some other sequence of nucleotide, 20 nucleotides, it will be called barcode 2. And those we are going to add to specifically each and every sample. So what we have is 96 bar-coded samples with us. Then we just mix them together and we call it as a library. So library is made of books. So what is our books? Barcoded genome is our book. Because ultimately we are going to write alphabets there. So we can call it as a library. That is why it is called as a library rather. Then pooling of sample and cleanup. Cleanup, why do we need cleanup? because we need DNA which is sufficiently long to identify. Small pieces are just useless for identification. So we'll have to remove them. And there are methods of cleaning up that particular DNA. We have magnetic beads to which, depending on the pH, that particular DNA gets attached. We can determine a larger, longer DNA or a smaller DNA. Generally, smaller DNA are removed. Then adapter ligation. Now, you can tell me what is this adapter ligation? You'll have to stick it somewhere near that particular pore, the motor protein and all. So that is how we do it. And then we prime it. What we do in priming? We just pour it on that particular trough, which has that membrane on it. That is priming. And that is exactly what you are going to do as hands-on. This is that particular flow cell of nanopore which you are going to use. Those are the different things which you will be taught. And if you see that sensor array, that sensor array is a beautiful piece of electronic device. It has nanopores 1500 on it. And each pore has a micro electrode, positive and negative, on either side of the pore. And the whole thing gives electric current which is read by the machine. So preparing such a masterpiece is a difficult thing, and therefore they are charging us like anything. I hope you get the point. Try to just think of it. You have 1500 odd nanopores which are proteins. Right? And this nanopore has on one side, the upper side, an electrode, and the lower side another electrode. It's going to count the potential difference between that particular two electrodes. And such you have nanoscale electrodes. So that is the beauty of the whole thing. This is the software for bioinformatics. This is how it looks. It's a live thing which goes on, sequencing. The green thing which you see is a nanopore which is sequencing. So if you count all those squares there, they are going to be the number of nanopores on that particular disk. On that particular. There's something here. Try to run it. I think I've done it. This is a live thing going on. So if you see the number of pores which are sequencing, it shows exactly how many pores are being sequenced. Sir, I have showed it on mobile first. So you can have access to it through TeamViewer or any desk, and you can see it at your home. You can make a run here and then see whether it's running properly or not. Yeah, and ultimately when the sequencing run, you get enough data, maybe 5 GB of data for a 48 run or 96 sample run. You can stop it and then ask it to base call. Now can you tell me what is this base calling? Base calling. Base is ATCG, electric current, base call. Which software is used there? Guppy. ते नाव जेव्हा ऐकलं तेव्हा मला जरा हसायला आलं होतं कारण गप्पी मासे माहितीच आपल्याला. Right. So this is the workflow, sequencing workflow. I'm moving faster. Fast 5 files and fast Q files. I have already dealt with them. These are the raw data, the zeros and the ones. Fast 5 file. It's the hierarchical data format, and it is read by the machine only. This is a typical fast Q file. So you have a header there. Then the actual sequence. You can see there G, C, A, G, G, A. That is the sequence, actual sequence. And below that, after a plus sign, what you're seeing there are the ASCII characters which are showing the quality of each base. So something written below that G, F. That F is telling you how much is the quality score. So for each base, you get a quality score, and then you can combine the score and see whether your run was proper or not. Whether it had enough depth or not, enough coverage or not. Enough coverage means you have 30,000 of them. If in that 30,000 you are not picking up 15,000, your coverage is bad. And such sequences are not taken by GZ. They say you have 50% Ns. Ns means nucleotide which are not identified. So they say that Ns तुम्हारे 15,000 से ज़्यादा है, ये तुम्हारा लेने वाला नहीं है, माफ करो. So that is how it's the coverage. The coverage has to be good. And depth, वही sequence, वही nucleic acid, कितने बार तुमने read किया? तो जितना ज़्यादा read करोगे, उतना तुम्हारा confidence बढ़ेगा, quality score बढ़ेगा, and you are confident that this is the nucleic acid which I have identified as A. 99.99% times it was A, and therefore I am confident that it is A. So quality score is high. It is counted in something called as the Fred score. If you see, it is 10, 20, 30, 40, 50. And generally, all sequencing devices tend to see to it that their score is at least Q20. Now you'll understand why it has to be Q20 from the next graph. See the graph. If you see very carefully, after the Fred score of 20, before 20, the whole thing is very steep. So you are totally having no confidence of whether your base was correct or not. But after 20, it's almost a straight line. So even if it is 30 or 40 or 50, it's not making much of a difference if you are able to cross Q20. So that is the ultimate goal, to get at least to Q20 for each and every sequencing machine. So let me tell you here that the Q score of the oldest archaic sequencer, Sanger sequencing, on which the whole of human genome was sequenced, has the highest Q score. The later advanced technology Illumina has a Q score lower than Sanger. And the latest one, which is called fourth generation sequencer, Nanopore, has it still lower. So as your generation has increased, your basic Q score is reduced. So what they need to do is, they need to go in for more consensus. So more and more sequences should come in, so that the average then comes to be a good Q score. Right? Individual Q score उसका Q7 तक ही है. But then when it goes to consensus, they can reach Q30 also. Am I complicating the things? I'm sorry for that. Okay, PAN files. These are again the files which this software alone knows. Then there's a benign software which is inbuilt in the machine. It would be shown to you. I think you have already seen it in the video. FP2E platform, this is also free of cost with Nanopore machine. It identifies the variant. So this is FP2E, it's free of cost to one who purchases it. One who purchases it gets it free. Otherwise you don't get it. This is how it's identified. If you see at the top there now, it's 21L I suppose, and it's Omicron. That has been identified by this software. This is the FASTA file. See very carefully, it is just those 4 alphabets there, nothing else. When VCF, it's a variant called format file. Bioinformatics simplified is free of cost. It's free of cost only for SARS-CoV-2. You do something else and you pay for it. Pay for it through your nose. It's pretty expensive. Next slide, I told you, 22D, please don't forget it. It's ours, right? So don't forget 22D. This is the next slide there. This is how we identify. This is how next slide identifies all the variants. You see Omicron there. The first one it has identified as BA.2.75. So it combines Pango also. This is how you can go in for phylogeny, lineages. The lineage of your variant. And this is the one for molecular aggregation, where you get where is that particular isolate of yours. You have got it from whom? how it has spread, the thing which we discussed is this one. So if the same person is giving it to the other, it would be on the same node. On the same node, means at the same point. The mutations would be identical. So we can catch hold of it. These are the people who work on it. Pangol is the next one. This is that website. And it identifies in such a way. Identifies in such a way. And this is how it shows the distribution. You can download the CSV file. CSV file tells you what variants which are there according to the numbers. It's a comma separated. Usher is also very important. Usher was very important for us as such for BJ because we could identify BA.2.75 with the help of Usher. Because Pango and Nextlayer were a bit slower to identify. Usher was very fast. Fast means they adopted those mutations for identification methods. This is how it identifies it. It also tells you about the next strain. Next strain means its immediate neighbor. Immediate neighbor. So again important for phylogenesis. Again important for molecular epidemiology, where you come to know which is the next neighbor of it. Next neighbor, it can find it out for you. So for example, the same strain which is found in India, found in US, tells you that the virus took a plane and came there. The person took a plane and came. WGS, this is a very important website. This was initially made for influenza, but now it's also there for SARS-CoV-2. We have uploaded, I think, if I remember correctly, maybe something between 1,500 to 2,000 genomes of this. This is Indian initiative where we upload on WGS we upload the fast data, and on INDA we upload the fast Q files. That is how we do it. Something about Illumina, I just made a fast reference to it. Somebody from Illumina will tell you more about that machine in the afternoon. This is their workflow. These are their machines. Of these, this department has a MySeq. So MySeq sitting just in the center of all. Right. So that machine we have. Iron torrent is the instrument of NIV. They are using iron torrent. This is I told you about this, that H ion changes. So H ion is identified. This is their flow cell. I had shown you the flow cell of nanopore. This is the flow cell of iron torrent. I don't have it. I'll have to go to NIV for it. I have the Illumina flow cell which I will show it to you. This is the whole thing. Hydrogen ion which is released is that particular electrical change is calculated. So you can see that. That is how they identify the nucleotides. Thank you. Free for questions only, fast questions. Thought I have
Sir, we would like to request you to explain how the sample runs through the nanopore membrane and how it stops.
Yeah, it's the favorite of my students. Yeah, it's the favorite of my students because I was explaining it to them in my room, where I was telling them that ये हिल को देखो इधर हिल को देखो, ये जो खिड़की है खिड़की वो एक पोर है, तो उसमें से वो जाएगा. And all those things, they are just referring to that particular thing which was in a very colloquial language.
No, that would make us clear exactly how the membrane.
Yeah, if you have a membrane, if you just don't have those adapters and all, you don't have the ligands and all, just try to understand that you have a large wall on which there are small holes there. What would happen is when you put a sample there, it's not necessary that the sample has to go through the pore. It would go and bind anywhere. It would just go on the part where you don't have a hole, and therefore your sequencing run would then be very long. So that's why you need all the steps which will be told to you as the time goes on of why to attach certain adapters to it, so that it's specifically taken. Ligands are there which drop in near that particular pores, and they are the ones to which that particular strand will stick later on. So you are putting something which is sticky on two ends, one near the pore and one on that particular sequence. So it comes near the pore and then goes to the pores exactly. So your time is reduced. Otherwise, by chance and Brownian movements, everything would go to the pore someday. But then it won't be as fast as it is now. It is just 72 hours you can get a result, or even less than that you can get a result. So you won't get a result. But so उधर जाके बस वो कहीं जाएगा नहीं.
Colors capture.
Yeah, it helps to capture by telling that the pore is here.
Just like oiling.
Like?
Oiling.
Yeah. It doesn't go very fast there. Yeah. It also, but then it teeth is there. Teeth are पकड़ के लाता है. Again?
For any sequence, absolutely good.
Yeah.
Not to report it
Yeah.
on the software.
Yeah.
How does it go about?
No, we have special softwares for that, which tries to find a quantic. To find a quantic, what we do is they try to search in for a specific sequence and then place those sequences together. In that way, they get a quantic. Like for example, एक टुकड़ा छोटा सा है, 8PCG, उतना 4 का ही है. दूसरा है ए कुछ भी और 80CG फिर से आ गया और आगे कुछ और है. तो ये 80CG जो सेम आ गया, उसको एक साथ ले जाएंगे. और ऐसे 100, 1000 ऐसे जब करेंगे तो you will get a pattern where उसको सेंटर में ला लिया आपने और फिर बाकी भी उसके अगेंस्ट और भी आ रहे हैं उसके साथ. तो ये सब टुकड़ों को वो एक साथ करेगा और एक अलग अलग क्वांटिक्स को लेकर के एक जीनोम फॉर्म करेगा. Right? It's a software which does this. It goes on doing it. Because that question is very obvious that should come to you. Because Wuhan strain का sequence निकाला कैसा? वो तो comparison के लिए कुछ था ही नहीं उसको. So that is the work which the Chinese scientists have made. The Chinese लोगों को गालियाँ देते हैं, लेकिन they were the ones who found out the original sequence and gave it to the world, so that world could come up with RT-PCR tests. They could come up with sequencing. So that was a good thing which they did. कैसा China bashing चलता है, but I am not a China favorite.
Science doesn't allow it.
Huh?
Science doesn't allow it.
Yes, science didn't allow it. Science punishes. Science is punished. Right? We had that experience how science is punished. Yeah. And Peacock got it. No, Peacock. The one who found out Omicron. What happened? He told the world that I have found Omicron. The next thing which happened was all flights to South Africa will stop. They had a huge loss of economy. So telling science is bad for economy. Right? ऐसा है. Yeah, it comes in play. Science comes at the last. Yeah. Anything
So
more?
where does the microarray techniques stand in genome sequence?
Microarray
Array.
is a very primitive technology. Go on. Microarray चलेगा सर. ये तो whole genome sequencing है. Microarrays में तो एक particular part of DNA पकड़ते हैं. It's a technology of past. Microarrays. वो तो लोग तो करते हैं कि microarray, लेकिन microarray is a technology of past. Genome sequencing, वो genome sequencing है. जब तुम टुकड़ों में करोगे ना, तो वो microarray हो गया सर. जब तुम पूरा की पूरा लोगे, तो वो WGS हो गया. And WGS is always better because microarrays में तुम identify वो कर रहे हो जो तुमको मालूम है. जो मुझे मालूम है वही identify होगा. WGS में तो पूरा sequence निकलेगा. So it's an advanced technique. WGS is more advanced than microarrays. हाँ. और कुछ? Yes sir. The youngest student is asking a question. Yes sir. The youngest student has the last thing to get up. अच्छा comment करने वाले हैं.
It's a complimentary comment to Dr. Karyakarte's simplification of this very complex technology of genomic sequencing. Now I would say that why people should understand genomic sequencing. Anybody will ask a question. There is a community medicine person, and there may be a lay person who are producing machines. They may have a question in their mind. Now we have faced a situation where the mutants have arrived, and there was a thing which was earlier. There was when COVID came, we did not have drugs, we did not have any vaccine, we did not have ways to prevent it, and step by step things started flowing in, and we came to know certain things. Now let us understand this in the context of the development of immunity against the vaccination which has been introduced. Vaccination was introduced, Covishield or Covaxin or whatever was there, and our body has developed the immune mechanism to overcome the natural infection caused by the circulating cold virus. Now he has explained to us the importance of genomic sequences to understand the immune escape ways in which the mutants are causing infections, and there are some which are spreading very fast, like say for example B2.275, and he has predicted it will cause widespread infections everywhere, though of minor form. So from the language of community medicine, it is like from a highly virulent COVID, where there were a lot of first wave, second wave, not many deaths, morbidity and deaths were there. And as these 75 took over, the milder form of the COVID has come to exist. And you will find gradually the virus will go into endemicity. And in due course of time, I will not be surprised if there will be development of herd immunity. And if you understand the mechanism of this sequencing, whatever the four nucleotides we have mentioned, it relates to the T lymphocyte and the surface markers that we have got. And it's a lock and key mechanism which operates. And if you understand that, and I think the simplification which Dr. Karyakarte has offered us will get into our daily technology of our the grey matter. Even I have understood something, and I am sure that many of you will be your young minds. Mine is a aging and more senile mind, and I may not be able to grasp it as much as your young minds can have grasped. And I am very thankful to Dr. Karyakarte for this very exemplary exposition of this technology. And I am sure the day is long, and you will all be longing to await certain more discourses. Hearty compliment, Dr. Karyakarte. Thank you. And I am very well. Thank you, sir.